Good morning. If you have not already done so, please let me know that you are here in the chat. Hopefully it's working. All right. Let's get started. So, first of all, before we get into anything, I'd like to start the day off by asking people if they have any questions from the content from yesterday. So, anything? Anything you're not sure about? I'll take a moment and clear that stuff up here before we get started with today's stuff. It's pretty impressive. Almost all the muscle soreness is gone from my injection. That's pretty fast. It only took a day. Okay, doesn't look like there's any questions. If there is, I can always circle back. So, we started off the unit talking about, oh, my mic is peeking out all of a sudden. Why does it do that? We started off, weird. We started off with the unit talking about biodiversity. What kind of tools can we use to measure what's out there? And in order to measure what's out there, we have to establish how do we differentiate between things, living things. So we talked about a little bit about differentiating between living things, like sorting them into the idea of species, and then a little bit on why you should care about biodiversity, which is sort of kind of the thrust of the entire course. We're going to talk about the idea of diversity in every unit, and then that is all supposed to connect back to day one, which is, and also care about it because it's important for our survival as humans. So that's sort of how the curriculum is designed here. But anyway, it's all about diversity and increasing resilience and survival. And you'll see that theme come up many, many times in the course. So what we're going to discuss today is the idea of classification. So there are so many different things out there. How do we keep them all straight? We have to have a good system in place for organizing living things. If we want to monitor biodiversity, if we want to keep track of how things interact with other things, we need a good system of classification, an organizational system for life. So one thing to quickly mention here at this point, oh geez, I lost my train of thought. Okay, never mind. Let's mornings, am I right? Everybody had their morning drink of choice. The name of that science of organizing living things is called taxonomy. So taxonomy is the science of organizing living things. There's a lot of stuff out there. Probably about 9 million species in total. Is it just me, or is this just like a tiny little square today? What's going on with this? Why is it teeny? There we go. That's better. Where was it? Oh yes, taxonomy. There are about 9 million living things. That's an estimate, obviously. We haven't counted them all, and we certainly haven't identified all of them. So what that estimate is based on is, it's approximate. There are other estimates out there, but they're sort of in the neighborhood of 9 million. But the idea is we've identified a certain amount. I think it's like 3 or 4 million. And we also, every time we look, we find a certain number in certain areas. So every time we survey the ocean floor, we find this many new species. Every time we survey grasslands, we find this many species or whatever. Aquatic makes up a huge portion of this, by the way. The majority of living things actually live in the ocean. That's how this number is assessed. Okay, so based on that, we can sort of get an idea for approximately how many are left to be identified. There's about 9 million. So there's some statistical analysis going on there. And yeah, we got to keep them straight. You guys may have previously talked about other types of organizational systems for living things. In grade 9, you would have talked about the idea of classifying things as producers, primary consumers, secondary consumers. So there is some value to that, to classifying things based on what they eat. But that is not a robust enough classification system for 9 million things in order to keep them straight. And, oh, this is what I was going to mention earlier. There are organizations, scientific organizations, that are responsible for doing this process, this classification process, and different groupings of species have different organizations that are responsible for the identifying and naming of things, like the International Association of Mycologists, who are in charge of naming and classifying fungi, or there is a similar organization for plants, and there is an organization like that for vertebrates. And there are different groupings that have like sort of their own organization that's responsible for the naming. And the reason why they're international is our taxonomic system is a international system. We try and keep it consistent. So if you're researching, whatever species of field mouse in the plains of North America, and someone else is researching a different species, or maybe the same species, there is some crossover between continents, in another country on the other side of the world, you're talking about the same thing. It's the same species, so the characteristics that make you classify that species as that species are consistent worldwide. And when we do that, we provide them with a name. So we're going to get into the naming stuff in a second here, but this just is like a little side road here. We give organisms a two-part name. That two-part name is called the binomial nomenclature. That's a two-part name. This comes up again later, by the way. But just important to note that when you're writing a two-part name, a binomial nomenclature, it is always italicized if possible, keeping in mind that obviously if you're handwriting, it's not possible to italicize. But if you're typing it, you italicize. You capitalize the first name. That's the genus. Okay, as I was saying, you italicize the name, you capitalize the first name of the two-part name, and you don't capitalize the second name, okay, which is known as the specific species epithet. You don't capitalize that part. So some of these that you may have heard of already, you probably have heard of, for example, our species. We are Homo sapiens. So that's how we would write it there. It's italicized. You capitalize the genus, which is the first name. And the second name is the specific species epithet, and you don't capitalize that one. You can also short form the genus, which is, again, the first name in the scientific name. And so you could just write it as H period sapiens. That's another way to write it. And you've probably seen some scientific names written that way as well. For example, like Escherichia coli. Escherichia coli is almost always shortened down to E. coli because nobody wants to spell Escherichia. In fact, I think that if you asked me to spell Escherichia right now, I wouldn't be able to spell it properly. I'm going to try it. Escherichia. Oh, this is wrong. Escherichia. Oh, was I close? Escherichia. Oh, I'm even pronouncing it wrong. Escherichia. Escherichia. There we go. That's the E in E. coli. Escherichia. So a lot of people short form that down to just E. makes your life a little bit easier. So anyway, those are the names that we give them, and they are international. I should also mention that every time we start naming some supposedly new things, whenever we bring them, awesome, thanks Owen, whenever we bring them in, to a new conference and say, hey, I discovered this, I want to name it this, the international organization has to review your the characteristics of your species and decide whether or not it actually has already been named. Because oftentimes they figure out, oh, hey, this is actually the same thing as someone else already discovered this and called it something else. So, and every once in a while they do inventories of all of the species. I read an interesting article on the BBC not that long ago about the International Organization of Mycologists, again, fungi people, getting together and doing an inventory of all of the names. And they found out that there was like 2,000 names that were doubled, as in they had the same species with 2 different names. So, so that does happen sometimes. And then they just needed to figure out which one they were going to keep and which one they were going to get rid of. So anyway, that's how that works. If you discover a new species, you have to bring it to one of those international organizations. And you're allowed, if you discover it, most of them allow you to propose a name. So some people name them after themselves. I think that's a little bit big-headed, but you can really name it. There's like a style of naming that exists, and you kind of get a feel for it when you see enough scientific names. It's like a Latinizing of the name. There's certain letters it has to end with, and like stuff like that. Then you get to propose a name, and if they accept it, then maybe you have a species named after you. So anyway, if you're into zoology or whatever, that might be a thing that happens one day. Lots of stuff left undiscovered. Anyway, where does the system come from? It's old. It's obviously we named things forever. We called this like sheep or whatever. You just we named the objects around us, but we didn't have a great organized system. For example, saying all of these things are plants, and plants do blank, and that's how you know it's a plant or whatever. That originated in the early 1700s, mid 1700s, with a scientist whose name was Carl Linnaeus. You guys are going to watch a little video later about Carl. Seems like a good guy. Pretty important in the classification game. And that's where we got our original system of taxonomy, how we organize living things. So this idea of the 2-part name, and then there are different levels of categorization that go down from the most general all the way down to the most specific, which would be species. So we're going to talk about those names or those levels of classification. So those levels are called ranks, taxonomic ranks. And so at every rank, at every level, you are given a specific grouping. Okay? And we're going to go through some groupings together in a second. Oh. I hear something in the background here again. I'll be back in 2 seconds, okay? Sorry about that. Okay, let's keep going. Okay, so let me take you through some examples here. I'm actually going to start with humans. Okay, so over on the left-hand side, you will see the different levels of classification. The very top level, up here, right here, is the domain. That is the most inclusive group, the largest group. And then as you go down through the levels, they get more and more specific as you go down. Okay, so if you can imagine, this is like this is sort of like a filing cabinet, I guess, where it's like a cabinet in 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 a cabinet. And then when you get to the very smallest one, you're getting to like the most specific place, the specific species that you could classify something as. Whereas the very top thing, the domain, is the broadest categorization that you can give to something. Okay, so I'm going to go through the classification, the ranks for humans, And then I'm going to go through some other examples, some other species, and show you what their particular ranks look like, so you can see what groups they're placed into. And this is all based on characteristics. So initially, Linnaeus' system was based on morphology. Okay, we've already talked about morphological species classification, the idea that you have to look at their characteristics to classify them. So initially, this entire system was based on similar physical characteristics, morphology, and a little bit behavior, but mostly physical characteristics. So if you can imagine, Linnaeus was going around and saying, okay, you are green, or, well, I mean, color is probably not a good way to go, but you photosynthesize, so I'm going to put you in this category. And you do this. You have 4 legs. I'm going to put you in this category. And you have a spine. I'm going to put you in this category. And so that was the initial system. It's based on morphology, and everything that had the same things went into the same groups. And we still use that system to this day. So you actually need to know what the taxonomic ranks are. Okay, that's part of this course, and this will probably come up in any other course that you ever take on zoology or microbiology or really anything. You do need to know what the ranks are. So the ranks go domain, kingdom, phylum, class, order, family, genus, and then the most specific is species. Okay, species epithet is like within the genus, that it's that specific epithet, that specific species. And then when you say species name at the bottom, it's the genus and the species. So that's these last two here put together is what makes up the binomial nomenclature for a particular species. It's the genus name and the species name. Okay? So you'll need to place that somewhere into your memory banks. Domain, kingdom, phylum, class, order, family, genus, species. That's the order that it goes in from most general to most specific. So domains are very broad. There are 3 domains of life. We're going to get into more detail with them later. But the domain that we belong to is Eukarya. And you may have just briefly discussed this in grade 10. You probably did if you were in my class anyway. Eukarya are eukaryotes. Anybody remember what a eukaryote is? Oh, actually, I have it written right there, so it's maybe not a great question to ask. They are organisms that have organelles, basically. Membrane-bound organelles. Mitochondria, Golgi bodies, that kind of thing. Okay? So Linnaeus actually didn't have this level of classification because he wasn't able to look at organisms under a microscope. So he wasn't able to see any of these things. But they also didn't even know bacteria existed back then. So the other 2 domains were basically unknown in Linnaeus's time. So we added that one later. But there are 3 domains. Eukarya is one of them, and it is everything with a membrane-bound organelle. So mitochondria, Golgi body, endoplasmic reticulum, etc. Those are all have membranes around the outside of them, the organelles. So we have that. So that means we belong in this very broad grouping. Now, of all of the eukaryotes, we are in a specific kingdom. So that's the next level down in the classification system. Okay, the next rank. So we are Animalia, animals. That's our kingdom. And how do you figure out if something is an animal? It belongs in our kingdom. Well, it's able to move on its own. So there's another word for that, a scientific word for that, and that's motile. Oh, whoopsie-daisy. It's a little bit too thick. Let's go smaller. Okay, move on their own is motile. Now, you only need to be able to move on your own during one part of your life cycle in order to be considered motile. So sea sponges are a great example of something that's an animal, but it's actually stuck to the ocean floor for most of its life. But when it's basically a baby sea sponge, it's allowed to float around in the ocean, swim around. It can swim to find a place to attach to the ocean floor. And so because it can move around during some part of its life cycle, we call that motile. So it's motile, and then the second component of that is it has to be multicellular in order to be an animal. Believe it or not, there are things that are motile that can move around that aren't animals, but they're all single-celled. So if you're multi-celled and you can move around, you're an animal. So that's how you establish whether or not you belong in the kingdom Animalia or not. In this course, we're going to spend most of our time hanging out at the kingdom level. So what we're going to do is we're going to go through all the different kingdoms of life and talk about how you fit into one of those particular kingdoms, the basic properties of each kingdom. And then in future courses, not necessarily in high school, mostly in university, if you're interested in specific phylum of those kingdoms, let's say you're like really into fungi, which is one of the kingdoms, well, you can take a course in mycology, the fungal kingdom. And in that one, you're going to examine the different phylums of fungi and then probably look into the different classes in each of those phylums of fungi and what makes them unique. Okay, so you get into more specific areas here of fungi, for example. And later in your university career, if you're really into a particular family of whatever, or order or family, you can often take courses that are specific even just to a specific order or family. For example, if you're super into primates, you can take some primatology courses. Order primate is a subcategory of the mammal class. And so this is like a specialization in zoology. Zoology is just the study of animals. So it's within the animal kingdom. And then, so you get what I mean. You get more specific sort of as you go up. If you have a particular interest, and then you can really focus in on a particular group. Cetaceans, if you're like into whales and stuff like that. No, there aren't courses offered on literally everything, but a lot of specific groupings have sort of higher academic study that you can get into, and usually you could become a scientist in one particular area if you're interested in the ecology of fungi or something like that. You could get real specific if you wanted, or insects or whatever. These things exist. So for us, we are a particular group of kingdom Animalia called chordates, okay, or Phylum Chordata. Phylum is just a subcategory of kingdom. So of all of the animals, we are chordates. And how do you establish if something's a chordate? Well, it has a spinal cord. I think I actually took this table directly out of the textbook. It says animals with a backbone are chordates, which is not correct. So there are actually lots of mistakes in the textbook, which is unfortunate. Every textbook has that. But there are lots of organisms that don't have a backbone. I shouldn't say lots. There are some organisms that don't have a backbone, but still have a spinal cord. And those are chordates still. So you don't have to have an actual bone in order to be a chordate. You do have to have the bone to be a vertebrate, but a vertebrate is actually a subphylum of chordates. It's a level below the phylum, but above the class. So that's another thing to mention here, is that these are the original levels, the original ranks of taxonomy, but we've added in extras. So we've actually added in additional groupings here below phylum and above class called subphylum, or sometimes they're called superclass. There are some above order and between class called superorders or subclasses, which can get a little bit confusing. So we've added in additional levels in order to sort of fine-tune the classification using the system. But for this course, I don't care about those at all, and neither should you. Okay, so you might see them in your research when you're looking up your specific species, but you don't really need to care about them. So for our purposes, these are the only levels that you really need to be concerned with. Okay, so just as a heads up, there are more levels that exist, but I'm not going to dig into it. So unless you're getting into unless you are very interested in getting into mycology or whatever, some specific, I don't know why I keep saying mycology. There's lots of different studies of organisms, but unless you're getting into a specific one, you don't really need to know those categorizations. I don't know them, and I dare say most biologists don't know them unless they're working in a specific grouping. So anyway, don't worry about those. So of the chordates, we have a spinal cord. Again, this is us that we're classifying. We belong to a specific class of chordate called Mammalia. And you probably already have an idea of what a mammal is. They are chordates that have fur or hair and milk glands. That's what makes something a mammal. So there are lots of mammals that also fit into our same class. Things like whales, cows, pigs, dogs, cats. A lot of the things that you see in your everyday are mammals. So I think in general people are very familiar with mammals, but we all share that in common. We all have fur or hair and milk glands. Of all the mammals, we belong to a specific order of mammals that has grasping fingers. Okay? So if you think about things in nature that have grasping fingers, and by the way, they're called primates, that's what a primate is. So primates are a very broad grouping that includes monkeys, chimps, gorillas, orangutans, civets. There's a lot. It's a pretty big grouping, and all humans, obviously. But really, anything that is a mammal that has a grasping hand, we would consider to be a primate. Now, they don't have to have an opposable thumb. So a lot of primates actually don't have an opposable thumb. They just have a grabby hand. But that is what makes something a primate, if you're a mammal and you have that. Of all the primates, we belong to the family hominids, or hominidae. All of the hominids are primates that have a relatively flat face and 3D vision. Okay? You might be thinking, well, what are the other hominids? There are none. So there used to be a number of hominids, which we're going to talk about actually in the evolution unit. We're going to spend a day on human evolution, actually, and we'll talk about what the other hominids were, where they went, what evidence we have for the other hominids, etc. But they all went extinct. So right now we are the only group within the family Hominidae. You might be thinking, oh, well, what about chimps or gorillas? Well, they don't apparently fit the definition of relatively flat face. It's because they have that protruding sort of snout on the front. I'm not sure if that snout is the right word, but so if you look at a primate skull and turn it sideways, you can actually see in the skull they have like a sort of like a protruding mouth area, which no hominids have. Like hominids have basically flat faces. We are hominids. I'll turn my face in profile here. You can see like it's a fairly flat face. So that's why we fit into this particular grouping. Of all the hominids, and there were many, we belong to a specific genus called Homo. We are, again, currently the only member of that genus. However, we were not always the only member of this genus. So I'll use Neanderthals as an example. Neanderthals were our closest relative that died out sometime around the end of the last ice age. And we were both part of the same genus. Okay? So the way you get into that genus is that you have an upright body position and a relatively large brain to body size ratio. And Neanderthals certainly had that. In fact, they were very similar to humans. They had what you would call culture, technology. They made arrowheads. They established complex social relationships. They had funerary practices where they buried their dead. They were very much like humans, culturally at least. However, there are some key differences. Again, I'm going to leave this for the evolution unit because we're going to talk about them in more detail there. But they're extinct, obviously. So we're still the only ones within that genus. And then lastly, our specific grouping within the genus is Sapiens, sometimes called the specific epithet. And what differentiates us from the Neanderthals and other groups is that we have a relatively high forehead and notably thin skull bones. That's our main difference between us and the Neanderthals, who have a much thicker skull bone and a relatively protruding forehead. Again, we'll look at that later. But I actually have some example reproduction skulls from a Neanderthal at school. I don't know if I can access those, but I'd love to show you when we get to the human evolution unit. So anyway, we have a common name as well. A lot of species have a common name, not every species, but many do. So we do as well, humans. And those are our various levels of taxonomy. And you'll notice that at each level there is a description. How do you fit into that particular level? Why are you in this family? Why are you in this genus? So when you do your project for your particular species, whatever you choose, you are going to create this exact chart, basically, where you're going to explain to me why your species fits into every rank of taxonomy. Okay, so just keep that in mind. You're going to be reproducing a chart very similar to this for your project of whatever species you choose, and it can't be a vertebrate. Okay, let me show you some other examples so you can see how we can potentially differentiate between species using this system. Okay, so I mentioned that the very highest level here is the domain, and all of the things that we're going to be discussing here are from domain Eukarya. So that's shown at the top here, the eukaryotic domain, which just means you have membrane-bound organelles. There are two other huge domains of life with many, many species in them. They are all single-celled life, bacteria and archaea, which we're going to talk about on a different day, but we're going to focus on domain Eukarya for now. So let's look at the difference between us and, let's say, dogs. Okay, so the modern domesticated dog. Well, we share some groupings with dogs. We belong to the same kingdom, obviously. We're both animals. We belong to the same phylum. We're both chordates. We belong to the same class because we're both mammals. However, after that level of organization, it starts to break down a little bit. So, I turn my mic down. I keep peeking my mic out here. Sorry. We belong to different orders. So we are primates, and if you'll remember, that's the idea of the grasping hands, okay? Whereas dogs don't have grasping hands, obviously, and so they have other characteristics that are used to put them into that order, carnivora. Okay, these are primarily meat eaters. There's something to the configuration of their teeth that places them into that grouping. Okay, and within carnivores, they belong to a specific family called canids or canidae. And in that family, they belong to a specific genus, the Canis genus. And within that genus, the Canis genus, we call them familiaris. So familiaris is just the specific species that we've assigned to dogs. So their full name would be their genus and their species, which would be Canis familiaris. That's the domesticated dog. Now, this is another example of a species that is established using the morphological species concept. So if you go back to the other day, we talked about the idea that in general we use that biological species concept, the idea that it's a group that can interbreed under natural circumstances and produce fertile offspring. But with dogs, we have had to make an artificial morphological grouping here in species because the gray wolf and the dog is the same. They're not really actually different species. I mean, we call them different species, but really, they're not different species. What humans have done is genetically engineered the gray wolf over, I don't know, the last 50,000 years, as long as we've been domesticating dogs, a very long time, by the way. And we've been manipulating their genome, but through breeding, to produce the modern domesticated dog. But any dog and any gray wolf can mate, and in fact do mate. If your dog runs into the forest, it could mate with a gray wolf, and it can produce offspring that are 100% viable. So this is an artificial distinction that we've created based on morphology. So, and that being said, we even classify all domesticated dogs into the same species. We call them all Canis familiaris. You can take a Chihuahua and you can breed it with a greyhound or a Doberman or some other ridiculous combination. Now, you may run into some problems in terms of the offspring fitting. Usually the larger of the two dog breeds is the female in that circumstance. Otherwise, the baby won't fit sometimes and will cause significant problems. But in general, you can interbreed any two species of dog. It's physically possible to do that because they are all the same species. So anyway, I just wanted to mention that. This is sort of like an artificial distinction that we've made here. So that there are two species that are relatively similar to each other. Okay, that means they have a lot of the levels of taxonomy in common. But we can look at another species like the fruit fly, which does not have very much in common with us, except certain things we do have in common. So for example, we are both motile during some stage of our life cycle, and we both are multicellular. So we're both animals, us and fruit flies and dogs. But that's where the similarities sort of end. They don't have a spinal cord, and we do, as do dogs. So they fit into a different category of phylum. And then of the arthropods, they belong to a specific class, Insecta, and then you can go through all the way. But in other words, we have fewer levels in common with them, which just means that we are more different than they are morphologically. Okay? Or we could look at something very different from us, like an onion, for example, which is Allium cepa. It says kappa in your textbook, which is also a mistake. There's no such a thing as Allium kappa. And we don't even share a common kingdom with them, with onions. They are not motile at any stage during their life cycle. They have a seed part of their life cycle, but that's not motility when you're just, because they can't move of their own will. So they belong to a different kingdom because they're very different from us. So essentially, the more of these groupings that you have in common with another species, the more closely you are related to it. And we're going to talk about level of relation a little bit later. That idea is called phylogeny, and we're gonna dig into that on a different day. But essentially, the more groups you have in common, the closer you are related. So this is the system of organization for life. It's called taxonomy. Before I move on, oh, and at the bottom here, I've just mentioned this. I already mentioned this, but this idea that there are levels in between these levels, but you don't need to know them for this course. Any questions about this idea of taxonomy and how it works before we move on? Whenever I ask a question like that, I think what I'm going to do is keep going. And then if anything pops up in the chat, then I'm just going to circle back just to keep things moving because there's quite a bit of a delay. Okay, so 9 million things. Maybe we've only classified a couple million, 3 million maybe. How do we keep them straight? Let's say that you're a scientist working in a field in Oregon, and there's another scientist in Alberta, and they're studying the same grasshopper, and there's lots of grasshoppers in the field. How do you know that you're looking at the same one? Well, you need some tools for differentiation. And that tool that you use to figure out what the heck it is that you're looking at is called a dichotomous key. A dichotomous key is a decision tree. Basically, you ask a bunch of questions about the organism that you have in front of you, and based on your answers, you identify what specific species it is. I mean, it's that simple. And every question is a dichotomy. A dichotomy means that it's one or the other. It's a choice between two things. Dichotomy. So something to keep in mind. You guys are going to be making your own dichotomous keys for the assignment as well. And you can't have a three-way choice where it's this, this, or this. It's got to be a dichotomy or a two-way choice. This or this. But there's nothing in between. Okay, so let's, and it's a tool that you can take out into the field, and you ask these questions, and based on your answers, you can identify whatever species. So just so you know, you can buy a dichotomous key for North American birds. Okay, you can just buy that. Look it up on Amazon or whatever, wherever you buy books, chapters. And so you can go out, find a bird, and then you look on the first page and it'll say, does it have this or this? And you look at it and say, oh, well, it has this. So go to page 5. All right. Does it have this or this? It has this. Well, go to page 10. And then you go through the dichotomous key, and by the time you get to the end, you have identified which species of bird it is. So it's quite a valuable tool for biologists because we can tell what we're actually looking at. And so going back to what I mentioned earlier, where there's these organizations, these international organizations of whatever, you name the species type, they usually are responsible for creating tools like this to help identify and ensure that everybody is discussing these same species. Okay, so let me give you an example of a very basic dichotomous key. This one right here is for identifying the type of invertebrate that you are looking at. So assuming that you know that you're looking at an invertebrate, remove all this stuff here. Which would mean that, first of all, the species that you're looking at is a eukaryote. That means it has membrane-bound organelles. Those are pretty easy to identify. And you know it's an animal. So it's motile and it's multicellular. So you know that. You're looking at that. And then you also can tell that it is an invertebrate animal. Okay, it does not have a spinal column, a bone in its spine. All right, so this is a dichotomous key to help you identify which specific type of invertebrate you're looking at. So you go out in your garden and you see, oh, of course, my iPad's going to conk out here. Hold on one sec, guys. Technology. Come on. Every once in a while, my iPad just gives up and just says, no. We're not going to do that anymore. Okay, you go into your garden and you see a little earthworm. Crawling around in your garden. And you want to know what kind of invertebrate is this earthworm. So first of all, you're going to make a choice when you look at it. Is this organism asymmetrical or symmetrical? So is there a line of symmetry within the organism? And hopefully from math class, you know what symmetry means, from geometry. So if there are two sides, if you could basically split this organism up, can you split it into sections that are the same? And if you look at something like a porifera, which is a sea sponge, no, you cannot. There's no way to split this up into asymmetrical pieces. If you look at this, excuse me, there is a way to split it up, which is that you can draw a line right down the center of it, and it's symmetrical on each side. So this is, in fact, a symmetrical organism. So you're going to go this way in the dichotomous key. Okay? And then you have to ask yourself a second question. So is the symmetry bilateral or is it radial? So bilateral symmetry is when you see something like this, where there is two sides. That's what bi means, two. So two sides that are symmetrical to each other. Or is it radial symmetry? And radial symmetry means that there are sections that come out from a center, and each section is symmetrical to the other sections. Okay, so for example, cnidarians, which include things like these guys, which are jellyfish, are radially symmetrical. Well, we look at our guy here. It's not radially symmetrical, it's bilaterally symmetrical. So we're going to go this way on our dichotomous key. And then we come to another question. Does it have an anus? And believe it or not, that is relevant for animals. Things like platyhelminths, which are flatworms, don't have an anus. They only have an in, no out. So their food goes in and then waste comes out the same hole. Worms, as you probably are aware, do have an anus. So they have a digestive tract that goes from the front to the back. So we're going to go this way on our table. And then, is there segments that are visible on it? Okay, if you look at mollusks like octopi, for example, there are no visible obvious segments in them. However, obviously, if we look at this, you can see visible segments in an earthworm. So we're going to go this way. And then lastly, does it have an exoskeleton, a hard carapace, or no exoskeleton? And obviously, worms do not have an exoskeleton. They don't have a hard outer carapace. And so we're going to go this way. And then finally, you end up at the end point, which is going to give us our specific classification. So these are annelids. Earthworms are a type of organism called an annelid. There's more than just earthworms in there, but they fit in this grouping, and they're not arthropods. They don't have an exoskeleton. So that's an example of a dichotomous key for establishing whether or not something is an invertebrate, or which type of invertebrate it is, rather. Okay, and you can design these keys for anything. This one next door here is for deciding whether or not something, or this is for deciding what type of vertebrate you're looking at, if it is a vertebrate. So let's say you go in your backyard and you see a robin, okay? Does it have fur? No. Does it have feathers? Yes. Okay, well, it belongs to this particular grouping of vertebrates, birds, okay, otherwise known as aves. Are you able to get the door? Okay, thank you. I lost my train of thought. Oh, right. Yes. So what we're going to do right now is we're going to create our own dichotomous key together for a grouping of butterflies. Okay. Hopefully you guys kind of get the idea how this works. This is the one that I made with my last class. I'll get rid of this. We're going to make our own together. So, some things to keep in mind when you're making a dichotomous key. You want your key to be as, first of all, you want it to be based on morphology. So, oh, sorry, question. What about the mammals that don't have fur? Marine mammals. Great question. Bree, that is a fantastic question. Can you give me 2 seconds here? I want to answer that question. I mean, I can tell you that they have mammary glands. I know that whales breastfeed their babies. It's a really interesting milk, actually. It's like extremely thick that comes out. But you're right, they don't have fur, and most mammals, and it's often cited as one of the reasons that something is classified as a mammal. So, give birth. They have fur, although it's very sparse. That's the first I ever heard of that. They have fur? This looks like a relatively trustworthy site. Huh. That's fascinating. I don't think I knew that they did have fur. No, that's not a good website. Hold on. Let me find something reliable. Whalefacts.org. Come on, give me something here. National Oceanic and Atmospheric Administration. That sounds reliable. They have hair. Honestly, Bray, this is fascinating for me. I did not know that. I've never touched a whale or anything, but apparently they do have sparse body hair. I'm fascinated. When you actually look at the evolutionary history of whales, killer whales and things like that, they actually evolved from a wolf-like species. So that's not really that surprising that they have some form of hair on their bodies. But anyway, that's a really good question, Bray. I don't think I knew that they did have fur, but they do. Anyway, you got to base your dichotomous key on morphology. So remember, morphology are physical characteristics, or technically behavior is okay here too, but things like color are fine. Things like about their body shape and what parts are visible on the organism or whatever. Okay, that's morphology, characteristics. What you shouldn't use are things like looks like a blank. So I often see these on people's dichotomous keys, like looks like a check mark, or looks like a bush, or whatever, you name it. And the reason why you can't do that is because not everybody agrees that it looks like that. If you say something looks like a check mark, or looks like my Aunt Gertrude, or whatever you use, not everybody is going to agree that that does in fact look like that. So you want to keep it specific to the morphology of the animal, or it doesn't have to be an animal, of the organism. And it has to be objective. That is, everyone must agree on this particular aspect of the organism. So it can't be something subjective. Subjectivity is probably the hardest thing to eliminate from dichotomous keys, and people are always using subjective stuff, which makes it impossible to mark them from my perspective, because I'm trying to use your dichotomous key, and you'll say, well, it has a long leg. Well, what does that mean? What's long? Is like a meter long, or is 2 centimeters long for this thing? I have no idea what long means. So that's totally subjective. So you want to always use objective things. Now, instead of doing that, so for example, don't use looks like a, and don't use big and small. Okay? What you could use instead of those things are things like circular. It has circular markings. Okay? That's legit. The markings on it are circular. Or instead of saying it's big, it's larger than 2 centimeters. Or its front legs are larger than its rear legs. Those are objective things that can be measured, whereas saying that it's long can't be measured. Or tail longer than torso, or something like that. Okay, so just keep that in mind, that you always want to base it on morphology, and you always want it to be objective, or as objective as possible, in order to make the key work properly. Otherwise, basically, two people going out into the field using your key would get 2 different results, and then the key is useless. So what we're going to do together right now is we're going to build a key for these 6 butterflies. So we're going to start with a general grouping here that includes all butterflies. Okay, that's the beginning of our key. Just like at the beginning of this key, it started with basically all invertebrates. Okay, so of all the invertebrates, that's our starting point. We're going to split them into groupings. So what we want to do is we want to look at our butterflies, or whatever, in this case it's butterflies, and decide what is a trait where I can roughly split this grouping in half. It doesn't have to be in half, but what I find for students is that it's the easiest way to go, is to start by splitting your grouping in half. So what's a characteristic that half of these have and half of them don't? And I actually am asking, so if you want, you put it in the chat. I'll wait. Okay, we can do circular markings. Hopefully this isn't going to block my voice too much here. Circular markings. Okay, so I would say circular marking, circular marking, circular marking. Okay, the other ones I can't spot a circular marking on them. Oh, actually, that's not true. There's one right there. Okay, so 4 of them have it and 2 of them don't. Okay, no problem. So let's do the split here. So you're basically going to ask a question. Does it have circular markings? We've got yes, it does, and no, it doesn't. Okay, so 2 ways you can go. So you don't need to include which ones go which way on your dichotomous key. However, I'm going to include it on here just to help us keep things straight while we're working. And you can include it if you want, but just keep in mind that you don't actually have to write which ones are going which way. But here I've got D, B, E, and F have circular markings. Okay, I'm just going to write it on here just to help keep things straight for me. I'm doing it in a different color so it's not confusing. So A and C go the other way. Okay, so let's finish off A and C and finish off that direction on the table here. So A and C don't have circular markings. How can I differentiate between A and C? What's the difference between A and C? What question can I ask to say, okay, A has it and B and C doesn't? And I'll wait. A little bit harder to get audience participation in something like this, but all right, Hannah. Oh, you've got some other ones coming in here. C has antennae. Okay, that's a nice obvious one. Now, I must say that A, in all likelihood in real life, does also have antennae, but I don't think it got picked up when I scanned it. But that's totally fine. Okay, so let's say it has antennae. Okay, I'm gonna go with yours here, Mohammed. Okay. So especially because it's obvious in this diagram. So has and does not have antennae. So we're going to ask another question here to differentiate between the 2 remaining and say, does it have antennae? And yes, it does. No, it doesn't. So yes, it does is C. I'll use a different color at the end point here. So this is an end point for our dichotomous key. If you get to that spot, you know it's C. And the same thing for the other one. No, it does not is A. So if you get to this end point, then no. Then you know that it's A. Okay? Now going the other direction here, of D, B, E, and F, we've got to differentiate again. How do I differentiate between D, B, E, and F? And let me erase my markings here, and I'll try and I'll highlight the ones we're talking about. So D, B, E, and F. How do I know the difference between those? How can I split them in half or into different groupings? Okay. Okay, Jasmine, I'm going to use that one. So B, you're saying the torso is longer than the wings. That's nice and objective. So is the torso longer than wings, or does it protrude past the wings? You could probably say that in a way that was a little bit more accurate. But does the torso protrude past the wings? So only one of them is yes. So I'm actually gonna put yes up here, and that'll give me a little bit more space. So yes is B. So now we've removed B from the grouping. If you get there, you're looking at B. And I'm going to remove B. So now we've got to differentiate between these three. So what's a way that we could differentiate between these three? And I might actually be able to use one of these previous ones. Body extends beyond the wings. Well, none of them have that. Oh, sorry, that's the same thing, Mila. Yeah, you're right. That's the same thing as what Jasmine said. Yep, for sure. How could I differentiate between these three? Two circular marks versus 4 circular marks. Okay. Okay. Number of circular marks. Sure. So here, I'm assuming you're saying, okay, this is the 2 circular marks versus 4 circular marks. Okay. Okay. So are there more than 2 circular marks? Oops. I forgot how to spell the word circular here. Ah, what just happened? Made my eraser too big. There are more than 2 circular marks. Okay, so yes, no. So there's only one that has more. That's F. So that our endpoint right here would be F. And then of the no's, now we're only talking about E and D. How do I differentiate between these last two? Having a good think on this one. That's fine. Okay, so this, okay, Aaron, interesting idea. Rounded top wing edges. So I think we're going to run into a problem here where I think some people are going to say this is rounded and this is rounded. Or you could say top wing edge could be this edge, rounded, this is rounded. So I think we're going to have to dial that in a little bit if we're going to use that. These are connected to the torso. Oh, I see what you're saying. That there is a section here on the tail, and that. So I'm. You know what? I'm actually going to use what you just said, Ivana, and I'm going to modify it just slightly. I'm going to say that there is a gap between the wings and the torso, a visible gap between the wings and the torso. Okay, so for that I think that you would say that, well, there's no visible gap between wings and torso. There's no, but here there is a visible gap. So I'm going to modify it just slightly and say visible gap between wings And torso, yes, there is. No, there isn't. So yes, there is E. And no, there isn't is D. Okay, so now that we've established this, and this is just like a real dichotomous key that you'd use, you could go out into the field, and then a butterfly would land on you and be like, oh, look at this guy. I wonder what it's called. You look over at your dichotomous key and you say, does it have circular markings on its wings? Yes, it does. Well, is the torso longer than the wings? Yes. Oh, well, that's species B. There you go. Functional key. Works every time. So as long as you've made your dichotomies objective, it doesn't matter who picks up your dichotomous key. They're all going to call that species the same thing. And again, that's really important because you need to coordinate scientists everywhere all talking about the same thing and calling it the same thing. So these systems are quite important. So then I'm going to propose an alternative question here. What happens if we add in a new guy? So you're out in the field and you pick up species, what did I call this? G. What do I do with G guy? Because this obviously happens all the time, right? I mean, you're going to go to your conference and say, hey, I discovered a new guy. G. Flappius butterflyticus. I don't know. Call it whatever you want. I mean, the genus is probably already established, but... I mean, you could throw the entire dichotomous key out, but that's a heck of a lot of work. That's not what we actually do. Okay, so you're on the right track here, Mohammed, which is, first of all, follow the path and see where it would go. Okay, so does it have circular markings? No. Okay, so we're going to go on the down here. Does it have antennae? Yes. Okay, so we're going to end up here on the key, but now we're going to have to expand the key from that spot. So instead of this being C, we're now going to have to add on some extra to our key. Create another dichotomy here. And we're going to have to separate between C and this. So have a look at C and that. How would I differentiate between this? Between C and G? Feathered antennae. Okay, cool. Cool. Yeah, that's a good one. That's a good one. Feathered antennae. Cool. Okay. Yes, it does. No, it doesn't. And so yes would be C. And no would be G. Okay, now we've added another species to our dichotomous key, and we can further differentiate and figure out what a specific species would be. Okay, and Mohammed, that one works too, for sure. Okay, and that's just what I wrote down here. You got to add an additional dichotomy. Okay, so what I'm going to get you to do, and what I'm going to do here is I'm going to do both lessons in a row. and then I'm just going to give you the rest of the time to work on the stuff from the lesson. The next lesson is a little bit shorter. And then I'm going to give you just all the stuff to work on at once here. But in a minute, when we're done the other lesson here, what you're going to be doing is investigation 1.2.1 on page 32 and 33 in your textbook. What that is, the first part, part A, is asking you to just read a key. Okay, look at how the key is made and read it. It has a written form of a key. If you look at it, it's a little bit different than these visual keys, where I'm drawing like pathways on a piece of paper. It's that one is called a instead of a visual key, it's called a oh shoot, I always forget the name of this thing. Anyway, it's a key where it's like a choose your own adventure novel, where it's just it's actually written out. Does it have this or does it have this? And if it has this, go to number 2. If it has this, go to number 3. So it's basically exactly like this, except it's just written out as opposed to drawn out. So in part A, basically, they're just asking you to use the key to identify some things. There's really not much that you have to write down for part A. It's just asking you to use the key and practice using a key to identify some things. Then in part B, you only need to do number 6. Okay? Number 6 is asking you to build a key from a number of organisms that are at the bottom of the page. So you're going to build your own key. In that case, it's probably easier to do a visual key. You can do the same written key the way it's done for part A if you'd like. It's totally up to you, but I find the visual keys much easier to make and understand, so I stick to the visual ones, but it's totally up to you if you want to do it the other way. And then you're going to try analysis questions A to C, but only A to C, okay? There's a reason why I'm only getting you to do number 6 and A to C, and that's because the other questions are not important for this course. So, I mean, you can look at them if you want, but don't worry about them. They're not particularly important, so I would skip them if I were you. So, anyway, that's where you're going to start. That's just some practice with dichotomous keys. And then there are some analysis questions that go along from the text. It's on page 20, numbers 5 and 7. So that's what goes along with this lesson. If you're ever looking for these on Brightspace, this stuff is always summarized on Brightspace, what you do and don't have to do. People often ask me, oh, do we have to do part C or whatever, or do we have to do this? It's always on Brightspace. So if it's not shown on Brightspace, you don't have to do it. Sorry, someone asked a question here. Is it in a real dichotomous key, could flying patterns be included? Yes, they could. So behavior is definitely incorporated into morphology in an actual dichotomous key. So things like hibernation behavior, mating behavior, calls for birds, all of those things are included as part of morphology. And so, yes, you do regularly see them included in real dichotomous keys. But that being said, when you're going to make your dichotomous key for me, this is for the project, please don't include behavior, because it would require me to research the behavior of every species that you're going to use in your key, because I'm not an expert in everything. And so if you could avoid using behavior, something that isn't immediately obvious from a picture, I would really, really appreciate that. Just because it makes it impossible for me to evaluate your key. But in real life, you could absolutely use behavior. So as I mentioned here, if you look, there's also a video that you're going to watch on taxonomy. This is an overview of how the taxonomical system works. So this is part of it. And then here it mentions again the questions that you're going to do. So it's investigation 1.2.1 on page 32 and 33, part A, part B, number 6, analysis A to C, and then the analysis questions, number 25 and 7. Did I actually? Yeah, that's right. Okay, and so what we're going to do right now is I'm going to go through this idea of phylogeny. We're going to talk about phylogeny briefly, and then I'm going to give you some time to work on all the homework questions together. So I better get moving here. So what phylogeny is this idea that, okay, sure, you can organize stuff by what they look like, but that sometimes leads to problems, because what we want in our taxonomical system is for it to be based on relatedness. And sometimes when you put things into groupings based on physical characteristics, it actually doesn't put things that are related to each other closely in the same category, because evolution's funny that way. Okay, so what phylogeny is taking genetic evidence, looking at the actual DNA of an organism, and comparing the DNA and using that DNA comparison to build your taxonomy structure. Okay, and usually it doesn't rejig the entire structure. What it usually does is it modifies it slightly. So again, perfect example. If you look at how closely whales and are related to each other, and you look at the blue whale and the killer whale, okay, orcas. Blue whales and orcas are not as closely related as wolves and orcas. Which, again, you look at that on the surface and you say, well, that sounds crazy. Orcas and blue whales look very similar to each other morphologically, and wolves and orcas look very different from each other morphologically. However, in actual fact, if you look at how related they are on a genetic level, orcas are more closely related to wolves than they are to blue whales. And that is because all whales, different groupings of whales, evolved to become ocean animals, mammals in this case, at different times. And they had different ancestors that led them into the water. And the blue whale's ancestor on land is quite a bit different than the orca's ancestor on land. So the orca ends up actually being more closely related to wolves than it is related to blue whales. I know, crazy, eh? So you can see how using exclusively morphology is problematic here. So how they do that, you don't really need to write this down, but what we do is we look at what are called homologs in DNA. So if you can imagine looking at the entire genetic code of an organism, all the letters of the code, and we'll talk about how the actual letter code works on a different day in genetics. That's on a different unit. But if you read out the entire code, you can imagine you can read the code. You can compare genes, sections of the code, to other organisms. Okay? How much of the code is the same code? And based on how much code is the same, you can tell how closely related you are to that other thing. Okay? And there's an assumption being made here, which is that all things are related to each other. And we're obviously going to talk about the evidence for that and how that works later. But just at this point in the course, you're just going to take my word for it. Everything is related. Okay? And we'll get into the evidence for that later. But let's say that you looked at the entire human genome and then you looked at the chimp genome. So if you were to take the human genome and compare it to any other human, your DNA, and compare it to the person who's in the house with you right now, or your neighbor, or whatever, okay? And you were to compare sections of DNA, so you're looking at how many genes are there, what do they code for, what you will find is that every human, well, almost every single human, has the exact same genes, okay? The actual specific code in the genes might be slightly different. Okay, those are called alleles. We're going to talk about this in genetics. But the genes are the same. You have a gene for this, your neighbor has a gene for that. You have a gene for eye color, your neighbor has a gene for eye color. Those are called homologs, okay? They do the same things. The genes do the same things. And your DNA is 100% homologous. Homologous means it has the same genes as every other human. Okay, we're 100% homologous to all other humans. We are the same species, so within a species, it's usually 100% homologs. If you look at species, or at humans and chimps, you will see that we are 98% homologs. 98% of our genetic material is exactly the same as chimp genetic material. Okay, we're 98% the same. So our level of relatedness to chimps is very high. Okay, we do have a common ancestor, and our common ancestor is not that long ago in geological time. Okay, it's not that many, it's several million years ago, but it's still not that long in geological time. When you look at how long life has been on Earth, it's not that long. Now, if you look at other primates, okay, like gorillas, orangutans, we have 96% homology to other primate groups. So it's not quite as high as with chimps. Chimps are our closest relative, But other primates have about 96% homology. That is, we have the same DNA 96% of the time. If you look at other mammals, like dogs, cats, whatever, they have roughly 95% homology to humans. And you might be thinking, well, a cat and dog, that looks really different than human. Yes, it does. The morphology is quite different, but the amount of DNA that is the same is very high. And when you really think about it at a biochemical level, what's going on in the cells, that makes sense. Because you have a gene to form eyes, and a gene to form your retina, and a gene to form your optic nerve, and genes that control how your pancreas works and your liver, and genes that control growth, and those things are largely all the same among mammals. I mean, a lot of the biochemicals that are in a dog, for example, insulin, testosterone, those chemicals are exactly the same in humans. They're like molecularly identical. So it does make sense that our DNA is largely homologous to other mammals. But then, okay, as you get further away, let's say that you pick something like a banana, okay? Now we're talking about something that is not very closely related to us and morphologically very different from humans. We still have 40% homology to bananas. That's insane. 40% of our DNA is exactly the same as a banana. that seems so crazy on its surface. But again, if you look at the biochemistry, we build Golgi apparatus. We have mitochondria. We have, I mean, and you name it. Like, pick all the different sort of cell organelle structures, some cell communication domains, some transmitters for intracellular communication. All of those things are shared between plants and animals. They're very similar because they're multicellular, so they have to have some similar biochemistry. And so 40% of our genes are actually identical. And then, of course, you can look to something that's extremely different from humans, like bacteria. Bacteria, obviously, very, very different. And we might even have 20% homology to some types of bacteria. And again, that might seem really crazy, but, we have the same DNA. It's made out of the same molecule. We produce RNA. We make ribosomes. We utilize those ribosomes in the same way. We have some of the same surface proteins. I mean, not a lot, but some of it is the same. And so there are going to be common elements between bacteria and humans. So, I mean, again, this must be mind-blowing for some people. We share common DNA with all living things, even bacteria. Okay, so we can use that level of relatedness if we look at homologs, which remember similar genes, similar sections of DNA, and we can use that to classify things, to figure out how closely related we are to things, and to modify our taxonomy based on genetic evidence. So again, that process of using genetics to modify taxonomy is called phylogeny, and that's sort of like the new wave of taxonomy. That's why I call it modern taxonomy, because we're using modern evidence to establish these things. So you'd be surprised how accurate exclusively using morphology or shapes or physical characteristics is at establishing how closely related things are. Linnaeus, who knew nothing about genetics, and genetics didn't even exist as a area of study at that time, but he still largely made groupings that were genetically homologous to each other, which is, again, very impressive. And obviously physical characteristics are related to genetics, so I mean that makes sense in some ways. But the original system of taxonomy is actually pretty good. It lines up with phylogeny pretty closely. Now, we do have to make modifications based on phylogeny, and by the way, there are many modifications yet to be done. So I'll give you a great example. We're going to talk about a kingdom called protists. Right now, based on taxonomy, based on morphology, we've categorized a bunch of different organisms together as protists. But if you look at their genetics, their phylogeny is actually very, very different. So So that's problematic. They probably don't belong in the same kingdom, and that kingdom will almost certainly be split up at some point in the future. Maybe by the time you guys get to university, protists won't even exist anymore. We'll have split it up into separate categories based on genetic evidence. Keep in mind that using genetic evidence like this is only maybe 20 or 30 years old. We didn't even have the technology to analyze the genetics of species until essentially my lifetime. So before I was born, this didn't even exist. There was no such thing as phylogeny. We didn't have genetic evidence. So that's kind of cool. So as I mentioned here, what you do with this phylogenetic evidence is you can establish a tree of life. So you look at all of the living species out here on the, imagine this is a timeline. You look at all the different living species out here, and you can connect them backwards and figure out where they're related to each other. And then work back. Oh, iPad, have you stopped working again? Sorry. Every once in a while, this thing takes a turn. Huh. Okay, so we can keep tracing it back. And we can build this sort of tree of life. Oh, that doesn't really make sense. You're not going to get that happening. But we build this tree of life where we can figure out when common ancestors existed between living things. And you can trace back the lineage all the way back to the very beginning of life, okay, on a timeline. There's no such thing as year zero here, but I just mean, we'll call that the beginning of life, which we call the origin. Okay, so the origin was the original cellular life on Earth. And yes, we can trace our lineage back like that with genetic evidence. And how we figure out where these splits occurred, these bifurcations in the tree of life, like how we figured out where they are in time, is based on how DNA naturally mutates. So we know how often DNA naturally gets changes in it, mutations. And so we look at the differences in our DNA between us and our closest relative. We look at the number of differences between us, and then we can get a rough estimate for how long ago we split off from a common ancestor with them. And so using that evidence, we can figure out, okay, well, we split off from the nearest ancestor chimps 10 million years ago or something like that. And we can keep doing that all the way back, figure out when we split off from plants or when we split off from protists or whatever. Okay, we can establish this complex tree of life, because keeping in mind that life has been around for about 3.8 billion years. That's a long time. And so there's been lots of opportunities for evolution to occur. We're going to get into this more in evolution. But anytime you see a bifurcation like this, which is a splitting of the tree of life, keep in mind that that is an event called speciation. It's when a new species was created, okay, through the process of evolution. And we'll talk about how that actually works, the mechanisms of evolution, a little bit later. Any questions about that idea of relatedness and phylogeny before we get into the specific phylogenetic tree of life? I'm going to talk about some of the actual categories on this tree in a second. But this idea of homologs and building a tree of relatedness, known as a phylogenetic tree. Okay, again, I'll circle back if anybody puts anything in there. I realize that there's a delay. Okay, so if we, again, here is an example of very basic phylogenetic tree of life. Phylogenetic tree is just a tree of life based on genetic evidence. And you'll notice that A down there, this is the earliest life, the origin. This is something that we have no evidence for whatsoever. I'm talking about what did the original life look like. There's no fossil record of cell life that is single-celled like this, and certainly not from this long ago. So we don't really know what the origin was like. We have some ideas of how the origin could have formed because we know a lot about what the early Earth's conditions were like. And experimentally, we can reproduce a lot of cool stuff in the lab using those conditions. For example, just creating an aquatic environment with the same components as the early Earth and temperatures and things like that, we can show that things like lipids, fats, can naturally form in the environment. RNA can naturally form in the environment. Cell membranes can naturally form in the environment, which is really cool. So we've been able to show that experimentally in the lab, but have we been able to actually create life in the lab using these early conditions? No, we haven't. So there are some pieces missing. We don't fully understand everything about the origin and how that could have formed. But we do have some good guesses in terms of the mechanisms behind the formation of some of the components of the origin, but just not how they came together. So there's quite a bit of mystery there surrounding the origin still. You'll notice that there is an early split here, very early in life. This is this phylogenetic tree does not have a timeline associated with it, but we're going to talk about timelines in a second. But one of the earliest splits here is between bacteria and archaea. So those are the other domains. We mentioned that there are 3 domains, or I mentioned that there are 3 domains. Bacteria and archaea are the first split of life. They are both single-celled organisms. They're both prokaryotes. So prokaryote is the opposite of a eukaryote. It does not have membrane-bound organelles. So there's no Golgi, there's no mitochondria, there's no nucleus. So bacteria and archaea do not have that, and they are the earliest split of life. And then there are obviously subsplits that fit into all the bacteria and archaea. And an offshoot of archaea was our early ancestors. So we're more closely related to archaea, and then somewhere later in geological time, domain Eukarya formed. And Eukarya includes all of your favorites, the classics: plants, animals, fungi, protists. Okay, they're all offshoots of domain Eukarya. So again, the 3 domains, this is the highest level of classification, are bacteria, which are single-celled organisms that are prokaryotic and have no membrane-bound organelles. Domain archaea, which are single-celled organisms that are all prokaryotes and have unique hazardous environmental strategies. That's what separates them from bacteria. They also have a different type of cell wall. We're actually going to spend a whole day talking about bacteria and archaea, so I think it's tomorrow, so I'm not going to get into too much detail there. And domain Eukarya, which are everything with membrane-bound organelles. So that is the highest level of taxonomy, the domains. And genetic evidence confirms this. In fact, genetic evidence is the reason why we created the domains. So the domains didn't exist before we had genetic evidence. We largely didn't really... I shouldn't say that. We had one domain for archaea and bacteria called Monera, but we recently, with genetic evidence, split Monera into archaea and bacteria. So there's an example of us actually using phylogeny, genetic evidence, to modify the original taxonomic system. Okay, I want to keep going here so I can give you guys an actual opportunity to work on this stuff. So, so what do you do with this? Okay, well, you can use this idea of relatedness along with morphology, and you can design something called a cladogram. So a cladogram is a phylogenetic tree. So it's a tree that shows relatedness, but what we've done is added in information about time. So that way we can figure out how long ago we shared a common ancestor with another organism. Okay, so that's called a cladogram. Cladograms show level of relatedness and time. So level of relatedness, or a grouping on a cladogram. So let me give you an example of a group on a cladogram. So if I can draw a line here. All right, well, I'm going to make it a lot thicker than that so you can see it better. Okay, I can draw a line around this, a common ancestor, and all of the species that are coming from that same common ancestor. So the common ancestor that I've selected is right here. And this is all of the species that come from that common ancestor. So that grouping on a cladogram is called a clade. Okay, what I've just drawn there is a clade. A clade is a grouping that has a common ancestor and every species that comes from that common ancestor. So we can use the study of clades to figure out how closely related we are to another species. So if we look here at this diagram, we are related to bonobos and chimps at the same level. We have the same level of relatedness to bonobos and chimps. So bonobos and chimps have a common ancestor, and we have a single common ancestor for bonobos and chimps. So we are related to bonobos and chimps at the same distance. Okay, we're as closely related to either of them. Bonobos and chimps are more closely related to each other because they form a clade. Okay? But they are the same level of relatedness to us. Okay? Because they have a more recent common ancestor in geological time, Okay? So if we looked at level of relatedness here between chimps and bonobos and us, okay, and then we looked at that same grouping but including gorillas. So this is a different clade that I'm drawing here, one that includes gorillas as well. We are more distantly related to gorillas than we are to chimps and bonobos. And that's because our common ancestor with gorillas is further back in time. Okay, about 8 million years ago is when we had a common ancestor with gorillas. So we are further related from gorillas than we are from chimpanzees and bonobos. All right, so that's the idea of clades here. Clades are built around levels of relatedness. And each of these circles on here represents what's called a speciation event. It is when one species became two species. And the idea of that concept of that is a little bit confusing. We're going to get into a lot more detail in the evolution unit, talking about how speciation actually works, as I mentioned. So don't worry too much about the processes involved in actually having a speciation event occur. We're gonna get in more detail there. Okay, so again, a clade is a single common ancestor and all of the species that are currently alive that are a result of that common ancestor. Okay, so this is an example of a clade. Now, I just want to point out, and I find this so cool, that the very first person to understand this idea that things are likely related to each other and that you can trace back common ancestry like a tree all the way to an origin was Charles Darwin. Okay, so this is Charles Darwin's claim to fame. That and natural selection, or evolution by means of natural selection. But if you look in Darwin's journal, this is so cool. I love this. It's capturing a moment of scientific discovery in Darwin's journal. Darwin drew out the first cladogram. He had been observing species that are similar as he journeyed around the world, and he drew this in his journal, which is the very first phylogenetic tree. I find this is so amazing. And then on top of it, so this is after he was like, well, I think that these 4 species are related, and if you go back to a common ancestor, they're related to these species, and B, C, and D, or whatever, are species that he's looking at. This is on his journey. Okay, he was specifically looking at island ecosystems for a lot of this. So he's looking at how different species from different islands are related to each other. But he, traced them back and said, okay, well, D looks like it's related here. And this is just from morphology, no genetic evidence involved here. But you can still draw these trees without genetic evidence. And then I love that in his journal he wrote above, he was like, I think. I think it works like this. This really seems like it would make sense. And so this is the essence of a scientific theory, which is I have a bunch of evidence, and I think this is an explanation for all of that evidence. So this is like a moment in history here captured, our first understanding of phylogeny, which makes my heart flutter. It's so exciting that like we actually have this like written down on a piece of paper in Darwin's journal. I think that that's really cool. So that's the first time that sort of arrived in the human consciousness, this idea of interrelatedness. Below here is a phylogenetic tree that shows various animal species. We're actually going to talk about phylogeny within animals a little bit later when we talk about animals. So I'm not going to get into too much detail with this, but here is a phylogenetic tree of the animal kingdom. So you look at the main splits here. These are mostly taxonomic morphological splits, but they line up with phylogeny, which is really cool. So this idea that within the animal kingdom, well, first of all, they're all multicellular. Okay, so everything in the animal kingdom is. There are ancestral protists that are also multicellular. They're not animals, but within animals, those are our ancestors. So the first split here is the difference between things that have tissues and things that don't have tissues. Sponges don't have tissues, but everything else does. And then the next split, and this corresponds to the split in geological time as well, is that you have things with radial symmetry. Those are cnidarians like jellyfish, and things that are bilaterally symmetrical. You notice that we talked about some of these distinctions earlier when we were looking at that tree of animals. Yeah, the Dichotomous Key of animals. So it was based on this. And then we have splits that are based on type of body cavity. Do you have a full body cavity on the inside with organs within it? Flatworms do not have that, but everything else does. And then you can look at collimates and pseudocollimates. That has to do with development of the digestive tract and when you're an embryo. And then of the collimates, we can look at the ones that are deuterostomic or protostomic. Again, these are terms you don't really need to know for this course, but anyway, we can go through and we can distinguish between all of these things based on morphology. So that obviously the original taxonomy here was based on morphology, and we can draw a phylogenetic tree based on morphology, but this is backed up by genetic evidence as well. Okay, so what are you guys going to do with that? How am I doing for time? I'm going to give you time, by the way, in the second block to work on this. So I'm not just, you don't have to do this during your break, which is coming up in a minute. But you're going to read tutorial 1 on page 22 and 23. So tutorial 1 goes through the process of reading a cladogram. Okay, I talked a little bit about cladograms here, but I didn't really talk in detail about how to read one. So it goes through the process of reading one in tutorial 1 on page 22 and 23. And then practice questions 1 and 2 are questions just about reading cladograms and level of relatedness between species. So you're going to try those questions on questions 1 and 2. It actually works through the questions with you in a solution. And then there are some analysis questions that go along with this. Again, they're all summarized on Brightspace as well. It's questions 3, 5, 6, and 9. It's written on Brightspace, in Brightspace right here. If you are interested in phylogenetic trees and you'd like to learn how to make a phylogenetic tree based on characteristics, there's a tutorial here, a Khan Academy tutorial, which shows you how to make a cladogram, essentially, from evidence that you've collected, or I should say characteristics, okay? Based on similarities and differences, you can sort things into a phylogenetic tree. You don't have to be able to make a tree for this course. You only have to be able to read a tree. But however, if you're interested in the process, or you want a more in-depth understanding of how the trees are made, this is a fantastic video. It's not required. I always throw extra stuff out there. If you're interested, I'll tell you if it's extra. But if you're interested, it would give you a deeper understanding of how to read the trees as well, because it's talking about how to make them. But you don't need to watch this video if you're not interested. But it's quite a good video. But I would watch the first video here, the taxonomy video, because it is going to go into detail on how we actually establish that kingdom, phylum, class, order, family, genus, species system of taxonomy, and a little bit more background on Linnaeus and what things were like before we established modern taxonomy. Okay, so watch this video. There are the questions that we mentioned about taxonomy and dichotomous keys. You're going to build some. And then the second part here is on cladograms. Now these questions are textbook questions, which means that they all go in your document, your question document for this unit. Okay, we're going to come back at 11:05, and I'm going to go through attendance, and I'm going to give you an hour to work on all of these problems. Okay, so you are going to get like a period to work on these, and then I'm going to come back and we're going to talk a little bit about kingdoms and domains, okay, in a little bit more detail. This is not a long note, okay, it's a fairly short note, and there is just like a short activity that you're going to do with that, but we're going to do it towards the end of class. Okay, so I'll put a time up and say we'll come back at blank time, and then you're just going to join me again. Okay, so I will see you guys at 11:05 for attendance. Please take a break. Okay, it's 10:20. You guys have been listening to my voice for a long time, and rest assured there is time for you to work on your own in the second half of the day. I won't be talking at you for the entire day. I'm sure it feels like a lot. But I'm going to let you take a break. Stretch your legs, and I will see you at 11:05. Hello. Welcome back. Please let me know in the chat if you are present for the second half. Sorry, guys. I got disconnected there for a second, it looks like. My apologies. I'm going to run a network cable over to where my laptop is here, because I think that the Wi-Fi may be a little bit untrustworthy. So as I was saying, I really appreciate everybody who participated in the first half. And again, if you're interested in going off course at all, asking a question, let's chat about whatever you're interested in. That helps keep the course interesting for you, relevant to you. And it's obviously it's harder to do like this, especially because there's like a delay in feedback from you, but you know, I'll fit it in where I can. So we've gone through sort of this idea of how things are organized. We talked about the morphological taxonomy, which is the traditional method, and then how we've now used phylogeny, which is genetics, to alter that original taxonomical structure, to add in genetic evidence. And we talked about some of the tools, dichotomous keys that are used in the field for identifying species. I realize that you haven't had any time to work on those questions yet. I'm going to give you that time in a moment. When we come back, so I'm going to go away, give you an opportunity to work on that stuff for about an hour. But when we come back, what we're going to do is talk just a little bit about what the kingdoms are. Actually, you know what? I'm actually going to do this right now really quickly because it's extremely short. It's only like 5 minutes long. And I may as well just do it now and then just let you work on all the stuff. And then you can ask questions as opposed to just coming back and doing 10 minutes at the end. That's kind of silly. So let me just really briefly talk about this. We mentioned the three domains: domain Eubacteria, domain Archaea, and domain Eukarya. So those are the three largest groups, the domains. Within the domains, so below the domains, there are 6 kingdoms. Okay? And it's a little bit weird how it's set up because domain Eubacteria only has one kingdom in it, bacteria. And domain Archaea only has one kingdom in it, Archaea. So that's a little bit confusing. So those two domains each only have one kingdom, and then domain Eukarya has all the other kingdoms of life: Protista, fungi, Animalia, animals, and plants. Okay. So overall, those are the 6 kingdoms of life. Okay. So they're listed down here. We have Eubacteria, Archaea, Protista, fungi, plants, and animals, or Plantae and Animalia. And as I mentioned previously, we are going to take some time now, pretty much for the rest of this unit, and go through and discuss each of these kingdoms in detail. And by the end of the course, I will be able to describe an organism to you, and then based on its characteristics, you should be able to go, oh, that's a protist. You're describing a protist to me. Or, oh, that's a fungus. That has all the characteristics of a fungus. That must belong to kingdom fungi. Okay, so again, these are the larger categories, not the domains, but just the next level below the domains. And we're going to talk about these in a fair amount of detail. So what I'm going to get you to do, this is the last piece of the work that you're going to do. And this does not go in your textbook, or in your, this does not go in your homework document because this is going to be in the notes. But you're going to use the table on page 27 of your textbook, and you're going to write down what are some of the basic characteristics that an organism would have in order to fit in each of these groups. Okay, so what does it take to be a eubacteria? What are the characteristics? What are the characteristics for archaea? And consider putting these into your own words. I mean, you can use some of the terminology that's in the text, no big deal, but consider paraphrasing what's in there, or thinking about what the actual characteristics are, and perhaps putting them in your own words. Okay, and then again, that'll help with memory and also with understanding the thing that you're reading. If you don't understand the words that you're reading, it's not going to be a terribly good characteristic to use to define eubacteria, for example. So just if you're not understanding a particular word, you gotta look it up or ask. Again, that's what I'm here for. And then for each of those kingdoms, you're just going to give me 2 examples, and for each example, you're just going to tell me what's the common name of that species and what's the scientific name. So keep in mind, not all of them will have common names. E. coli does not have a common name. Okay, that's a bacteria, by the way, that goes in kingdom Eubacteria. So if you just have the scientific name for something, that's fine, because there may be no common name. But in general, if you can find it, the common name and scientific name are what you're looking for. And also keep in mind that you're looking for actual species. So like for animals, you're not going to say goat or something like that, because there's actually a number of species of goat. And so you're going to pick a specific species of goat if you want to use that as your example. Okay, we mentioned some examples of animals previously already, like Canis domesticus. That's domesticated dog. So you could use that potentially if you wanted for a sample of animals. So this is just a very, very general overview of what goes into each kingdom, keeping in mind that we are going to look at these in great detail afterwards. So you don't need to write a novel for each of these, but what are the basic characteristics that would fit something into each kingdom? And then starting tomorrow, we're going to talk about those in detail. We're going to really dig in and look at what do the living things in that kingdom actually look like. Okay, I'm going to stop talking and let you work on the stuff for day 2. Again, it's all summarized here on the Brightspace page. So as you're going through the numbers, if we've done it already, cool. Move on to the next thing. The last thing I'll mention just very quickly, though, is if you haven't completed your student parent information survey, if you could please try and complete it today at some point, any point today before the weekend, because tomorrow I'd like to compile that information. And there is a practice quiz for day 1 and 2. There it is in Learning Block 3, sort of mentioned at the bottom. It says try the practice quiz. You can click on that. And it is just a quiz to test your knowledge from the stuff that we covered in day 1 and 2. How comfortable are you with how phylogeny works? What a clade is? Do you know the biological definition of a species? You know, that kind of stuff, okay? So you can try that practice quiz. Remember, the practice quizzes are totally for your practice. The idea is that if you do them and then you notice, oh, wow, I didn't do well on this particular thing. I don't really understand this concept. Well, that gives you a starting point to review your notes and come and talk to me during Learning Block 3. Okay, so I highly, highly recommend the practice quizzes. They will help you get prepared for the summative quizzes at the end of the unit. And if you're not doing them, it really works against you. So anyway, they're there for you. And the other thing I'll mention, the very last thing, is that as you're coming across new words while you work through this homework or readings or watching videos, don't forget that you should be adding them to your vocabulary list as you go. You can just add the words for now, and then during Learning Block 3, you go back and write your definitions, or you can do both. You can add that definition as you go, but probably do not leave them to the end. That has not worked out well for students in the past. So I wouldn't leave them to the end of the unit. Doing them daily and adding them into your vocabulary list is ideal. Before I send you off, is there any questions that people have in general about the content before you get into making your own dichotomous keys and assessing a cladogram? And I realize, by the way, that this is a lot of stuff. So this is normally a week of content in this course. But in order to meet the timelines of this strategy, doing a full course for a day, I have to take that information and condense it into a much smaller time period in order to make things work. So yeah, it's a lot. I totally understand that. I'm going to give you the next full, whatever, roughly 2 hours to work on it, though. Questions? One other thing just to mention is that each one of these notes or lessons that we're doing together is based on a chapter from the text. So you do have a resource there on taxonomy, dichotomous keys, phylogeny. These are chapters in your text, so you could potentially use that as a resource if you were interested. But yes, keep in mind, I am always available for questions. I will be here in the chat, working here. Essentially, I'm marking stuff for my other class, but I will be working here the entire time, and I will be there for if you joined me yesterday in the Google Meet, that's nice. Nice to see you and meet you. But I will be available the entire time between 1 and 2:05, and I'm happy to talk to you. Okay, I'm not seeing any questions, so I'm going to let you get to work. And again, let me know if you need anything.